Hello, everyone, and welcome to today's webinar, Efficient and Rapid Discovery of Novel Temperature-Sensitive Alleles with the Onyx Digital Genome Engineering Platform. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit inscripta.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Randy Lacey, PhD, Senior Scientist at Inscripta. Randy, you may now begin your presentation.
Okay, thanks for the introduction. So today I'm going to be talking with you all about the work I've done using the Onyx Digital Genome Engineering platform to identify novel temperature-sensitive alleles. So first I just want to start with a brief outline of the talk. First, I'll go through an introduction to temperature-sensitive or TS alleles in the Onyx Genome Engineering platform. I'll then go into the experimental approach I took and how I identified and validated novel TS alleles. Lastly, I'll conclude where the project's at thus far and where we're hoping to take it in the future. So starting with introduction to TS alleles and Onyx. So what are TS alleles? TS alleles are mutations in essential genes that allow for growth at permissive temperatures but restrict growth at non-permissive temperatures. So on the right here, I have a pretty simple example of a yeast spot plating assay where the top panel shows growth at a permissive temperature, and when that temperature is shifted to a non-permissive temperature, you get essentially no growth. And this is due to a single point mutation in the yeast genome. So TS alleles are very effective tools that are widely used in academic settings and also have use in industrial settings. In academic settings, TS alleles are function as genetic tools for studying essential genes in many different organisms. So I have a couple examples here I wanted to share of publications that TS alleles were central to. So on the top panel, this is a publication in PNAS from 1971 in which a TS allele in a fruit fly, in the cell cycle, a cell cycle gene of the fruit fly, was shown to have very substantial effects on the behavioral function of fruit flies as temperatures increased. So this is 1971. Fast forward 50 years to another publication in PNAS that was published earlier this year, where a TS allele was critical in understanding the function of, or how methylation is involved in ribosomal function. So I chose these two because they're both high-profile publications separated by 50 plus years of research, just to highlight that TS alleles have been relevant for a long time. If you dig through the literature, you'll find many high-profile publications where a TS allele was critical to the findings of that study. So in industry, TS alleles have a slightly different function. So in a bioindustrial context, we're always looking for new potential bioindustrial tools to help aid the goals that we're trying to achieve. So in the context of TS alleles, a specific example in which they could be useful is in small molecule production. So oftentimes organisms like yeast and E. coli are used are used for production of small molecules, and the production of those molecules are often linked to central metabolism. Central metabolism has several essential genes associated with it. So you can imagine during small molecule production where intermediates of central metabolism are shunted toward the production of a compound of interest, a TS allele could function as sort of a control switch, allowing for that intermediate to instead of continuing through central metabolism, it can then be shunted toward production of whatever compound that you're interested in producing. So this example on the right is from a publication by Amaris in Nature in 2016, where they engineered the yeast genome and inserted an exogenous production pathway for the production of farnesene. So you can see it's tightly linked with central metabolism, and there are several points throughout this pathway where essential genes are critical to the ultimate production of farnesene. So incorporating TS alleles in this pathway could help to increase farnesene production. So hopefully I've convinced you that TS alleles are important in both academia and industry. And so now I'm going to shift to essentially the problem with TS alleles, and that's that they're very difficult to identify. So essential genes are, because they're essential, they're difficult to modify, and many mutations will cause lethality in the organism. So TS alleles have been used for many decades, and the sort of traditional original methods involved random chemical mutagenesis with just massive phenotypic screens. And so this was effective in the early days of finding TS alleles. However, it's a very low throughput and time and labor intensive process. More recently, a method known as diploid shuffle was developed in yeast, and so the outline of diploid shuffle is seen here on the right. And so basically this is an error-prone PCR-based random mutagenesis method. And so there have been a few TS allele collections that have been generated from diploid shuffle, so it is a effective method for identifying TS alleles. However, it does have limitations. The first being a starting strain that is locked in, that you have to use for using diploid shuffle. So there's not much there's not much space to use different strains of yeast in this context. The second main limitation is you end up with a point mutation in an essential gene that is flanked by selectable markers. So there's quite a bit of genomic modification going on here that could have alone have potential physiological effects that might make it difficult to identify the specific effect of your TS allele. There have also been predictive methods, so computational methods that have sort of varying efficacy, but these methods are largely limited by the existing pool of TS alleles. So all of this begs the question, what are alternative tools for generating these useful tools in biology? So we know that TS alleles have relevance throughout many different areas of biology. So how can we more efficiently identify them? So that now brings me to the Onyx Digital Genome Engineering platform. So Onyx is essentially how I went from having never used TS alleles in my research and had very little knowledge of them to identifying many novel TS alleles in only a short period of time. So a few months at most. So now I'm just going to walk you through the basics of the Onyx platform, and then I'll go through a sort of example of what an Onyx workflow looks like. So Onyx is a benchtop platform, as you can see here, and it allows for scalable and precise genome engineering in both yeast and E. coli. So part of what's so appealing about Onyx is the simplicity of the workflow. So essentially, all you need is an overnight culture of your yeast or E. coli strain of interest. You put the overnight culture in the instrument, press start, and within a few days you have a highly diverse, highly edited pool of cells as the output of an Onyx run. Another unique aspect of Onyx is this covalently linked guide RNA, donor DNA, and barcode DNA system that drives the genomic editing, but it's also highly trackable with the DNA barcode. So you can follow the sort of progress of your specific edit designs throughout the lifetime of an experiment or project. So now, looking at the just a basic example of the Onyx workflow. So Onyx allows for iterative cycles of the design, generate, test, learn cycle of synthetic biology. And it starts with design. So the Onyx Inscripta engineering software allows for designs in both yeast and E. coli. So edit designs in Saccharomyces cerevisiae, you can have up to 6,000 designs per library in Saccharomyces, and you can have up to 10,000 designs per library in E. coli. and these designs can be quite varied. So you can make 1 to 63 base pair edits, and these can be insertions, deletions, or substitutions. So once you've settled on your specific design library, the next step is to actually build it. All right, so or to generate it, and this is done on the actual Onyx instrument. So after your build, you end up with a pooled engineered cell library, and so from this cell library, you can then go directly into phenotyping. So, and the phenotyping strategies can either be screening or selection approaches. So with your engineered cell library, you can also sort of assess the makeup of the library using a series of NGS assays that include both a barcode diversity assay and 2 different whole genome edit detection assays. And so these give you an idea of the diversity of edits, or the diversity of designs present in your engineered cell library, and also the fraction of those cells that have the edit, the intended Onyx edit. So as you're going through this test, the test stage of the design, generate, test, learn cycle, so you'll be going through, you'll perform your screenings and selections, and the next step is to actually learn from them. And so you can learn from the screening and selections using the Inscripta Resolver software. So Inscripta Resolver just takes the NGS data either from your engineered cell library or after screening and selections, and provides an analysis of the makeup of your cell libraries. So that completes the cycle, and then now you've learned about how your edits have behaved when you've applied specific selective pressures from your screen or from your selection. So from there, you can use that information to iterate again through this cycle and learn more and more each time you go through. So at this point, I'm going to shift gears and focus on how I used Onyx to identify novel temperature-sensitive alleles. But before I do that, I just want to briefly mention that Onyx is a very powerful and versatile platform that's not just for identifying TS alleles. It has very broad use in research communities, and it's particularly powerful in the context of strain development, so for optimizing small molecule production and protein production, and just in general in genome discovery. So identifying those underlying hidden aspects of the genome that we otherwise wouldn't be able to study or understand. So now I want to merge back from Onyx into TS alleles and sort of present the basics of how Onyx provides advantages over methods like diploid shuffle or similar methods to that. So the first is that Onyx allows for very specific trackable genomic edits with very minimal modification. So you only have the intended edit that you're interested in, and this is on essential genes. You get it from Onyx. You get your engineered cell library, which is just a pool output, and this can go directly into screens and selections. And you can also make a variety of edits and tags. So it's not just single point mutations. If you think that a specific tag can have a temperature sensitive effect, you can use that as well. It's also efficient in both yeast and potentially in E. coli. So you're not limited by a particular Saccharomyces cerevisiae strain. You can generate TS alleles in any of your cerevisiae strains of interest. So just to wrap up the introduction, I want to go over the main project goals. And so the first is pretty simple. It's just to identify novel TS alleles in yeast and E. coli. Using the Onyx platform to do this. And so this presents new tools in the form of novel TS alleles for both academia and industry, and also highlights the effectiveness of Onyx in this context. The second is to identify potential trends in TS allele loci. So from a first pass of identifying novel TS alleles, I want to be able to identify potential trends in where those TS alleles are located and learn from that, and develop a more potential rational design strategy for identifying TS alleles in the future. Okay, now I'm going to walk you through the experimental approach I took to identify these novel TS alleles using the Onyx platform. So the first step in this process is library design. So I've designed four different libraries, 3 in yeast and 1 in E. coli. The first 2 yeast libraries target 1 gene each. So the first targets dihydrofolate reductase, which is involved in folate biosynthesis and has no known TS alleles. The second targets HEM13, which is involved in heme biosynthesis. So the next two libraries targeting yeast and E. coli are related to one another. The yeast gene targets 10 different essential genes that have nine of which have orthologs that are essential genes in E. coli. So I'm hoping using these 2 libraries to identify trends trends in TS allele loci that correspond across kingdoms. So loci that produce TS alleles in yeast and also in E. coli. So central, a key component of each of these libraries are these internal positive TS allele controls. So in yeast, I have 3 different controls that produce published TS alleles. And in E. coli, there's 4 different positive controls that produce published TS alleles as well. So from here, I'm just going to focus on the yeast libraries. I've not gotten as much work done on the E. coli library yet, but I'm hoping to get to that in the future. But for now, I'll just focus on the yeast libraries. So the edit types that make up each of these libraries are pretty important. So DFR1 and HEM13 libraries are somewhat similar. They both have about 6,000 designs in them. Half of those designs are predominantly single amino acid substitutions, which you can see how that edit works down here with the examples of edit types. The other half are triple amino acid substitutions. And again, this is 3 amino acids replacing 3 amino acids. The rest of the library is composed of stop codons, synonymous edits, and those internal TS allele controls I mentioned before. And so you can see that there's about 200 internal TS allele controls, and the way that I populated out the 3 designs to 200 is using a variety of PAM spacers that are going to actually create these edits. The 10 gene libraries is slightly different. There's about 480 single amino acid substitutions per gene that target each gene, no triple amino acid substitutions, and then about 30 stop codons and 30 synonymous edits, and a reduced number of TS allele controls so that I could have a bit more single amino acid substitutions that are actually targeting the genes. So this is the design. I've now designed each of these 3 libraries. Now the next step is to actually generate or build these libraries on Onyx. So make the libraries, produce an engineered cell library, and begin understanding what the makeup of the population of those edited cells is. So that's what this data here on the top shows: the pooled edit fraction for each library, the pooled edit CV, and also the isolate edit fraction. So for each library, the edit rate is above 50%, which is really strong editing. The pooled edit CV is somewhat skewed for these libraries, and that was expected. We are making edits in essential genes, so there's going to be lots of designs that are likely to drop out, as these are going to be the lethal mutations for the essential genes that they're targeting. So we can also look at the actual design representation, and that's what this plot is showing down here. So these represent each of the actual barcodes that are associated with the designs, split up into the type of edits that those designs are supposed to make, and then plotted as log 10 design representation. And so each of these designs are hovering around 0 for log 10 design representation, aside from the stop codons, which makes sense. They should, in theory, be depleting, as these lots of stop codons are probably going to have lethal effects when they target these genes. So overall, these are really great editing and diversity metrics. And each of these pool of cells is now can now go into the test phase. So how can we start testing these libraries and applying selective pressures to pull out interesting edits that are potentially making TS alleles? So what we're looking at here is just a basic flow chart of how I perform the selection strategy. So I have 3 biological replicates where I have a sort of a pre-growth of a specific amount of cells at 25C. And so the actual number of cells is important here because we don't want to bottleneck our population. We want to make sure that we're starting with a diverse number of designs that can all be exposed to these selective pressures, and we can hopefully pull out information about them at the end of the experiment. So throughout the selection process, we'll have an initial pre-growth at 25C. This is then divided into 25C, 35C, with the remaining amount of these cells going to DNA extraction with barcode sequencing. We then grow these to saturation, which is about 6 generations, and repeat this process for generation 6, 12, 18, and 24. And at each of these points, we're taking samples for DNA extraction and barcode sequencing. So what we're doing here is sequencing the barcodes and getting an idea of how they're behaving over time at each of these time points. Are they enriching? Are they depleting? Or is not much happening to them? And from there, we can get information about whether or not they're actually behaving similar to known TS alleles. So now that we've tested our Onyx libraries by applying selective pressures, the next thing we want to do is start learning from that. So what happened to our designs over time as they grew at different temperatures? So to do this, we take those barcode read counts and essentially look at how they increase or decrease over the life of the experiment. And so we analyze these barcode read counts using this software package called Enrich2. So Enrich2 is a software tool that can provide statistical analysis for count-based experiments, similar to what we're looking at here. So the main output of Enrich2 are scores that indicate whether or not a design enriched or depleted in the population over time. So what we're looking at down here is the basic output of Enrich2. So each of these plots represent one design where each point is the log ratio that corresponds to read counts of that specific design, barcode read counts of that specific design over time. And the bottom axis is time points and generations. So looking at this first design here, not much happened to it. So the slope of this design is essentially 0 at this point. So its Enrich2 score is 0. Didn't enrich or deplete. On the right, we see that this design very specifically depletes over the course of the experiment. So it gets an Enrich2 score of negative 2.19. So this design depletes. You also get lots of statistical analyses like p-values, false discovery rates, and things like that can help you sort of have confidence in these Enrich2 scores that are produced for each design. So this plot that we're looking at here is all of these Enrich2 scores that you get for every design in a specific experiment plotted against the log 10 p-value. So you can see, all right, what's the Enrich2 score? And is that score actually statistically significant across biological replicates? So you get a pretty interesting spread of how all of the designs behaved at specific temperatures throughout the life of an experiment. So now let's get into the actual data from our libraries, so from the DFR1, HEM13, and 10 gene libraries. So each of these plots are just XY plots showing the x-axis with the 25°C Enrich2 scores, and the vertical axis is the 35°C Enrich2 scores. And so this is each design in the entire library. And so these are all the average. Each of these scores is the average of 3 biological replicates. So just looking at these plots, you can see for most of the designs, most of them have scores that for both temperatures hover around 0, right? So that's not entirely unexpected. That a lot of these mutations aren't going to have a substantial effect on how these designs behave throughout the course of the experiment. So what we're interested in particular, are designs that have not much of an effect at 0, or not much of an effect at 25 degrees C, so they're kind of hovering around 0, and they're depleting at 35 C, right? So sort of we're looking for designs that are in this fourth quadrant here, and so these are mutations that are likely causing temperature-sensitive alleles. So in order to start identifying these, or identifying putative TS alleles, we use the internal known TS allele controls that I mentioned before. So just a reminder, these are the actual mutations down here, and each of the colors here correspond to the designs that are the internal TS allele controls on each plot. So the first thing that stood out to me when looking at this was how similar across each of these experiments these known TS alleles behave. So this provided a lot of confidence in our actual selection strategy, because these are all independent selections performed at different times. So as I mentioned before, we want designs that are hovering around 0 at 25 C and are depleting at 35 C, and you can see that the MGE1 and ALA1 designs are very strongly depleting at 35 C with minimal effect at 25 C. HSP10 has a bit of a lesser effect, so TS alleles they're very sensitive to the conditions of the experiment. So they can behave like TS alleles in certain contexts, but not others. So it's not entirely surprising that this is not quite as strong of an effect as the other two designs. So this was a great first step, but we're just qualitatively looking at how these designs are behaving. What we want is to actually extract quantitative information so that we can start predicting what are these other designs that aren't known TS alleles that are sort of behaving like known TS alleles, and how can we quantitatively decide which ones to start targeting for potential validation. So here again, the internal controls are functioning as a guide for screening putative TS alleles in each library. So what I did was just a simple calculation where I took the score, I subtracted the 35C enriched 2 score from the 25C enriched 2 score for all of the designs that are making each of these internal controls, and average that value, and then I did the same thing for all of the designs that are targeting our genes of interest, whether DFR1, HEM13, or the 10 gene library. And so what these values show is that there's a sort of they're mostly strong positive scores, whereas the actual target edits aren't having quite as much of an effect, which again makes sense visually when looking at the plots. So with these values in mind, I wanted to next start to see which of these target edits are actually behaving at least similar, if not better, than all of our internal known TS allele controls. So I filtered out all of the target edits from each library that had a 25C minus 35C enriched 2 score of greater than 3. So better than average for all of the internal controls. And these are the numbers that we pulled out. So for DFR1, there's 85 designs that meet this criteria. HEM13, there's 188. In the 10 gene library, there's 123. So what this does is it provides a starting point for screening this TS phenotype. So these can be, with this many of these designs might not actually be TS alleles, and we might lose some. There might be some with scores less than this that are TS alleles, but this is a good starting point. And so we also, with these designs, we want to also take into account the statistical significance. So make sure that these aren't just random hits that fall into this category. These are actually significant and likely to be functioning like TS alleles. So just to actually qualitatively visualize how these hits look when plotted. So as expected, these are all of the hits. So each of the green dots are one of those hits that I just mentioned. And so they all fall into this fourth quadrant, sort of hovering around 0 at 25 and depleting substantially at 35. And so this becomes a bit more visually striking when comparing volcano plots between 25C enriched 2 scores compared to 35C enriched 2 scores, where you can see each of the green dots, which is one of these putative hits, is very, very much depleting when you show these plots at 35C compared to 25C. So this is I thought was a really striking representation to how each of these designs are behaving in this pool of cells throughout the lifetime of this experiment. Okay, so this was essentially finding, screening, identifying what designs are potentially behaving like TS alleles in these libraries. So now the next step is to actually validate these. So determine if these are in fact producing a TS phenotype. So there's a couple different ways to approach this, and I'll get into that when we get to the conclusions. But the first step I wanted to take was just to manually recreate a few of these sort of top hits from each of these libraries. So this is, I just wanted to, it's a kind of a convoluted process for reproducing these TS alleles that we identified. So this is just the process for individual validation of these TS allele hits. And this is in progress at this point. So the first step is to choose what I think were the sort of top hits that meet the score criteria and the statistical criteria as well. And so use these cassettes that contain our guide RNA, donor DNA, and barcode DNA, transform those with the Inscripta plasmid into our Saccharomyces cerevisiae base strain. Pick colonies, screen them for the actual presence of the edit, so make sure that the edit is in the colonies we screen. And then the next step from here is to cure out the onyx plasmid from these cells. So we want only the edit present in the genome. So the next step after that, you have an isolated, cured, edited Saccharomyces cerevisiae cell that has your potential TS allele. So then start screening it. So perform initial growth assays at permissive and non-permissive temperatures, and these can be in liquid, these can be on plates. There's a variety of ways to approach this. And then the last step is to actually confirm these TS phenotypes and explore the potential basis for what's driving this, the biological basis for what's driving this TS phenotype. So as I said, this is a work in progress. I've begun screening the HEM13, some of the top hits from the HEM13 library thus far using a spot plating approach. So here are the results from a spot plating assay, just looking at 6 of these sort of top TS allele designs. And so on the left we see a spot plating assay with growth at 22 degrees C, and then on the right is growth at 37 degrees C. And so of these 6 designs that we've screened thus far, we see two of these, so about 33% of the designs are showing a really strong TS allele phenotype. And this is super interesting, and we're starting to dig into the potential biological basis for this right now. So this is a really exciting point in the project. Okay, so the last section is just the conclusions and future work. So what have we learned thus far from this project, and where are we hoping to go with it from here? So the main conclusion at this point is that Onyx provides a very rapid and efficient platform for identifying novel TS alleles. So I'm still the actual validation is still a work in progress, but the hit rate at a 1/3 hit rate is pretty high, and so this is and only over like a couple month period. So this has been a pretty awesome experience to start identifying these novel TS alleles. So for future work, the biggest question we have right now is how can we increase the efficiency of identifying or of validating these TS alleles. So manually rebuilding each of these designs is pretty time intensive and laborious. So we're brainstorming ideas of how we can sort of increase the rate of this. So one idea we have is to actually make Onyx validation libraries. So from those 6,000 designs, pick the top 100 to 200 designs from that represent that represent putative TS allele hits, and then make another Onyx library that only has those. Build those designs on Onyx and isolate and screen colonies directly from Onyx, as opposed to going through the full selection experiment again. So we already know how these are behaving in that context, and then we can directly screen these on a plate or in 96 well liquid-based growth assays. So that leads into the second sort of the second component of this is optimizing these growth assays. So TS alleles are not on or off. It's not growth or not. There's oftentimes a intermediate growth effects that can be quite difficult to pull out when you're or to observe when you're screening many different isolates. So we're hoping to develop assays that are sensitive enough to pick up on growth defects that happen at non-permissive temperatures compared to permissive temperatures. We also want to perform the selections in E. coli. So the E. coli libraries, comparing them with yeast libraries, will be really interesting to see if we get similar hits, or if we get the hits at sort of conserved sites across each of these genes. And that leads into the final point of the future work is, and this is the second part of the project goals, and that's to identify or investigate trends in predicted TS allele loci. So this can help to aid in more rational design strategies. So I've started to dig into this data a little bit thus far. I noticed early on that many of these top hits that we've identified we're clustering to a small subset of amino acids in either DFR1 or in HEM13. So what I've plotted here is just a rank ordered amino acid position based on the number of predicted or of putative TS alleles that target that specific amino acid. And so that's what we're looking at for both of these plots. So for DFR1, over 50% of those 88 predicted TS alleles are target only 6 amino acids out of 211. And for HEM13, about 50% of the predicted TS alleles are found in only 19 amino acids, and HEM13 has 300 plus amino acids. So this is a very, very interesting observation, and really suggests that there are specific loci or specific residues that are more or less amenable to forming TS alleles. So the next question we have at this point is what about these residues are particularly interesting? So are they highly conserved, or are they not conserved at all? Are there specific structural features that define them that make these residues more able to form temperature-sensitive alleles, or their function? Is it something about the function of DFR1 and HEM13 that's driving this? And ultimately, as I've mentioned already, can these features inform rational design strategies for identifying TS alleles? So can we take information from this learning step of the cycle and allow that to inform the next sort of iteration of an Onyx experiment, where we can have more rational design strategies, where instead of having random amino acids throughout the entire protein, random amino acid substitutions throughout the entire protein, we have a few targeted amino acids that we know are going to be more likely to form TS alleles. So this is again a work in progress, but these observations are really interesting, and we're really hoping to learn a lot from them. So with that, I'd like to thank you all for listening, and I'll take any questions.
Thank you, Randy, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, if you wanted to find temperature sensitive alleles across many genes, how could you do this with the Onyx instrument with a limitation of 6,000 designs in yeast?
Thanks, Kaylee. Yeah, so this kind of gets at the sort of last point I was making there, in trying to identify rational design strategies. So I in the libraries I designed, I targeted a single gene and 10 genes. Um, and so we're hoping to learn more about where the TS alleles are located on each of these libraries, so that we can use that to make, let's say, a 30 gene library or a 50 gene library, where we don't have to do a random scanning amino acid substitution. So we can use that to target only, let's say, 10% of a protein with specific substitutions that are more likely to produce temperature sensitive alleles.
All right, thank you. Next question. What temperature are the cells at during editing on the Onyx instrument? Will some design edits die off if the cells are not maintained at the permissive temperature?
Yeah, so this is a great point. So on Onyx, editing and growth happens at 30 degrees Celsius. So this is, of course, an intermediate temperature to the two temperatures I was testing, both 25 degrees C and 35 degrees C. So with this in mind, it's definitely possible that this intermediate temperature could cause some of the designs that make TS alleles to sort of drop out of the population prior to me starting a selection experiment. But as I mentioned during the introduction, the actual growth during editing on the instrument it's a pretty short period of time. So it's from the start of the experiment to the end of an honest experiment. It's only a few days, and the actual process of editing is only a fraction of that. So it's definitely possible that some of these designs were lost in that process, but it's likely that most of them will be able to make it through the entire Onyx editing, or through an entire Onyx experiment to the end, so they can actually start and push them into a selection experiment.
All right, thank you. Um, Next question, there are actually two parts. Are the barcodes integrated into chromosomes after genome editing? And is this genome editing protocol with MAD7 scarless?
Yeah, so to answer the first part, no, the barcodes are not integrated into the chromosomes. The only thing that's actually delivered into the chromosome is your edit of interest. So the barcodes remain on the actual Onyx plasmid. And to answer the second question, is genome editing protocol with MAD7 scarless? So it's oftentimes it can be scarless, but there are instances where where synonymous edits need to be made in order to prevent recutting. So these would be very minimal modifications that just produce alternate codons. So that the wild type amino acids are produced, but there are very slight modifications surrounding your edit of interest.
All right, thank you. Next question. Why did you have to rebuild edits to validate temperature edits?
Yeah, so right. So all of these edits are actually being made on the Onyx instrument. So this is in the case of a selection experiment, it's a pooled assay, right? So all of the designs are mixed together in a liquid culture during a selection assay. So they're not separated. So in some cases, for some selection experiments, it would be possible to isolate those designs at the end of a selection by simply plating them and screening isolate colonies for the sort of hits that you're interested in. But this would require the hits from your selection experiment to be enriching, right? So so they have to be sort of increasing in the population over time, or having not much happen to them, basically. So I was specifically interested in depleting edits in this case. So these are going to be very likely, or very difficult to identify after plating the out or the end result of a selection experiment. So as I mentioned in the sort of future directions, what we're planning now is, so since we've learned from these first round of Onyx experiments to identify these TS alleles, we now have a general idea of what the potential hits are. So I'm planning to actually rebuild those on Onyx, as opposed to building each design manually. So build, let's say, 200 edits, plate those, and then screen and identify the hits from a sort of isolate screening design after an Onyx build.
All right, thank you. And we will wrap up with that question. Thank you again, Randy, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.